I'm Eric Nestler. I'm director of the Friedman Brain Institute, and it's a great pleasure to welcome you all to today's very important topic. This lecture is part of a series that we started almost 7 years ago now, which we call DiverseBrains, focused on diversity, equity, inclusion, and climate within the Friedman Brain Institute, the neuroscience community at Mount Sinai. And today it's my pleasure to introduce Sam McConnell and Astor Perkins. I don't know which of you is going to do the formal introductions, but I'll turn it over to you all. Thank you.
Thanks. Yeah, I'm doing introductions and Astor will be handling questions at the end. We're very excited to have Dr. Kale Edmiston with us here today. Currently, he is an assistant professor of psychiatry at the University of Pittsburgh, where his lab studies visual perception in people at risk of developing mood and anxiety disorders. He's the recipient of a number of awards, including the NIMH K01, the Brain and Behavior Research Foundation's Young Investigator Award, and early career awards from the American College of Neuropharmacology and the Society of Biological Psychiatry. And with that, Dr. Edmiston, I'll let you take it from here.
Oh, thank you so much for that very generous introduction. Okay, I'm going to go ahead and share my screen here. Is that working for everyone?
Yeah, that looks good.
Great. Okay, so yeah, I wanted to sort of situate myself in relationship to this literature a little bit before I dive into my talk. So I'm a neuroscientist. I use neuroimaging methods in my work. I've done neuroimaging methods for a really long time. So I identify both as a neuroimager, but also as a transgender person. And so the neuroimaging of transgender brains or transgender people is, something that kind of is at the intersection of two really important parts of my identity. And so that's why I've become, pretty interested in reviewing this literature and thinking about it pretty critically. Okay. Oh, also I should say, before I get started, I'm going to be reviewing some work that I consider to be pretty transphobic. And so just a heads up that there's going to be some transphobia, homophobia, even some garden variety sexism in some of this. So I just want everyone to be prepared for that. Okay, so where do we start? I think it's important to start with a very seminal study done by Simon LeVay, who is a post-mortem human tissue researcher. He is also a gay man, and he did a study in the 90s looking at volume of 4 different hypothalamic nuclei in the post-mortem tissue of a group of presumed heterosexual women, presumed heterosexual men, and then a group of homosexual or gay men, many of whom died of causes related to HIV/AIDS. So he found differences between gay men and straight men in the volume of hypothalamus and concludes from this, quote, "The discovery that a nucleus differs in size between heterosexual and homosexual men illustrates that sexual orientation in humans is amenable to study at the biological level." So I think that there's a lot of different ways that we can interpret this. And there's actually been quite a bit of science studies scholarship on this paper in particular that, if you're interested, I recommend you read. But I think, the most sort of innocuous interpretation we might make is just that it's possible to identify biological differences between these two groups. And that's basically what LeVay says in his paper. He doesn't say anywhere in the paper that he identified, what causes gayness. But of course, that's sort of how this study is remembered, for better or for worse. So this was done in the early 90s, 30 years ago. There's been a lot of progress for LGBT people since then. What has science done more recently. This was a paper published just in 2021. This was a large-scale GWAS, genome-wide association study, where they looked at a variety of different personality traits and behaviors. One of the behaviors they measured was same-sex sexual behavior. So the question being, have you ever had a same-sex sexual encounter? This is important because they didn't ask, do you identify as gay? What they found was that there was a specific allele that was associated with having had a same-sex sexual encounter in your life, and that the people who had had same-sex sexual encounters tended to have more opposite sex sexual encounters as well. And so from this, the authors conclude that they found a genetic marker that explains the evolutionary conservation of same-sex sexual behavior. If you read the paper though, this exact same allele is also associated with the personality trait of openness. So being open to new experiences, being, finding rewarding or finding novel experiences rewarding, that sort of thing. And so I think it's probably much more likely that what they identified was an allele associated with that personality trait, which mediated the relationship that they highlighted between the allele and same-sex sexual behavior. And so, of course, a lot of this nuance is lost when this sort of work gets discussed in public, right? This was sort of the headline for this study. That's, I would say, like, very different from what the study actually showed. And also, I would say that the authors made a lot of interpretive leaps in the paper itself as well. Oops. Okay, so I wanted to provide a little bit of context in terms of how people are talking about, gay people, cisgender gay people, because I think there's this narrative that we're always moving towards progress. And to my mind, the LeVay paper, although it certainly has limitations like every study, offered a much more nuanced interpretation of their data than this more recent study that was just published last year. So this is, now I'm going to talk about the Zhao et al. Study from '97. This is the first study of transgender people's brains that I'm aware of. This is also a post-mortem human tissue study. They looked at the number of somatostatin cells in the bed nucleus of the stria terminalis in cisgender men, cisgender women, cisgender gay men, and a category that they call transsexuals, and then also transsexual men, but it's actually a sample of trans women. And what they found was that the cell count of somatostatin neurons in this part of the brain were similar, that cisgender women and transgender women showed similar cell counts in this part of the brain, and that the two male groups showed similar cell counts. And so they conclude from this that they found a neural correlate of gender identity. They also say in this paper, they put forth the hypothesis that transness is caused by a prenatal insult in the brain that causes the brain to develop in an atypical manner, not consistent with the person's sexed body. I think taking these results and then making that leap to this sort of prenatal hormone hypothesis is pretty irresponsible. So I wanted to talk a little bit about what we know about somatostatin neurons and the function of the BNST. Okay, so the bed nucleus of the stria terminalis is a critical component of the anxiety circuitry. We know that it's involved in fear-based learning and perception of threat, the startle response, and it acts in a circuit whereby there's a fast processing of potential threat by the amygdala, and then and there are projections from the amygdala to the BNST, which is shown here, this tiny little blue part. And then the BNST then functions to filter and gate that information to be projected to the rest of cortex in order to mount a behavioral response. So at a more sort of molecular level, what does this look like in terms of gating and integrating? So in a healthy microcircuit between the amygdala and the BNST, at baseline, in the absence of threat, the somatostatin neurons function to gate pyramidal cell output. And so at baseline, when somatostatin positive cells are firing, there is no output from the PN cells. When there is a stimulus, a threatening stimulus input, however, we see less firing of the somatostatin cells and more firing of the pyramidal cells. And so what this creates is a relatively high signal to noise ratio, whereby we're seeing a filtered and processed signal to the rest of cortex in the context of a threatening stimulus. In a pathological microcircuit, where there's fewer somatostatin cells, we're seeing less gating of this signal from pyramidal neurons. And so what we have at baseline is an output that looks more like the output in a healthy circuit during fear. And so the idea here is that there's low signal to noise ratio in this instance, such that people are responding at baseline as though they are being presented with a threat. And so this is the sort of a theoretical basis for the role of somatostatin neurons in the BNST in the pathophysiology of anxiety. And indeed, we see lower somatostatin cell counts in anxiety and depression. And we also know that in rodents, chronic stress reduces BNST somatostatin cell count. So taking this, going back to this graph, we see these group differences, but we really need to be careful about attributing an observed group difference to a complex social identity, because there's lots of other explanations, because the environment for trans people is much more stressful than it is for cisgender people, all other things being equal. We also know trans people have a great deal of chronic stress. There's a 90% lifetime prevalence of major depression among the trans population. And so we're really comparing apples to oranges here. So why do scientists study transgender people's brains? I think that the literature tends to fall into one of three sort of buckets in terms of how the authors explain their motivation for studying trans people. I think the most common one is that they are interested in understanding why people are transgender. I think that there's a lot of variation and, very valid sort of opinions about the importance or usefulness of this question within the trans community. Some people think that this is, an important and worthwhile question. Other people do not. And I'll just leave it at that. Another reason is that they explicitly state that trans people are here functioning as a model organism for studying hormone effects on the brain. So doing longitudinal studies of trans people once they, before and after they start HRT, to help inform broader understandings of the function of sex steroid hormones. I think, again, I question sort of the ethics of this framework because trans people are a marginalized population that tend to be over-researched. Using trans people as a model organism to generate data that is not helpful to them in any way. I think, I would be willing to hear out arguments for why this is okay, but my sort of gut is that I don't feel great about that. And then finally, can we identify a biomarker for transness? So I think this is really borrowing heavily from the biological psychiatry world, where we're always talking about searching for biomarkers for things like anxiety, depression, bipolar disorder, schizophrenia. But of course, being trans is not a medical illness or a diagnosis. And so I get nervous when people talk about finding a biomarker for trans identity because of the history of medicine gatekeeping transgender people's access to transition-related care. I think it's not a far stretch to imagine some distant future where, a number of studies identified some biomarker associated with trans people, and then that is somehow used in a gatekeeping process. And, that is very frightening. And, we don't want that. So now I'm going to actually dive into the neuroimaging. So I'm going to divide this up by type of study. Obviously, there's a lot of different modes of neuroimaging. And I'm going to summarize the data overall from these studies, and then I'm going to pull out papers that I feel sort of represent a lot of the pitfalls with this literature. And then I'm also going to highlight some papers that I think have done a good job. So first off, regional brain volume studies. Since that 97 postmortem tissue sample study, there's been huge advances in MRI. And one of the main advantages of using multimodal MRI is that we're able to identify brain structure and function in awake human people. So when we do brain volume studies, these are using a high resolution structural image to look at group-based volumetric differences in specific regions of the brain. Most of these studies use a whole brain approach, meaning they don't have an a priori hypothesis. And so they're looking at the brain overall as a whole, comparing a trans group to a cis group. So as of 2021, there are 11 of these volume studies. They typically use a VBM method, which is an automated method. Major problem with these studies is that they all have fewer than 30 people in them. And the imaging world has evolved quite a bit in just even the last few years. I would say, I think there's a broad consensus that a study with 30 people in it is just grossly underpowered. And I think that results in how mixed the literature is. So literally in these 11 studies, every cortical and subcortical region of the brain has been implicated in trans identity. And I think that that's due to the lack of power, but it's also due to a lot of inappropriate statistical correction. I realize that talking about false discovery rate correction is perhaps not the most thrilling thing in the world. Unless you're a neuroimager, then you're obsessed with it. So if you're doing a whole brain study and you don't correct for multiple comparisons, your likelihood of having a false positive finding is just so astronomical. And some of these studies, they have an uncorrected p-value of 0.05, and they're comparing every voxel in the brain. So there's just no way that those studies would get published in neuroimaging journals. And that's why a lot of them are published in sort of like sexology journals. There have been no repeated findings, no replications. Nobody has found the same brain region twice. And the findings themselves in terms of the direction of them have been really mixed. Some have found congruence based on gender identity. So cis women and trans women having similar brain volumes. Some have found that assigned sex at birth is congruent and that there's differences based on gender identity. And then others have found that overall trans people of all genders have intermediate volumes between cis men and cisgender women. I'm not actually going to highlight any of these studies because I just don't think that there's a lot to say about them given how underpowered they are and just the sort of negligence with regards to statistical correction for most of them. Another structural technique that we can use is cortical thickness. So this is an automated method that allows us to measure the depth of cortex. And I want to highlight in particular one study done in 2008 by Woods. This study, again, really small sample, 15 trans men and 14 trans women. But one strength of this study is that they looked at, it was a longitudinal study over the course of 6 months. So they scanned these individuals at baseline, they went on HRT for 6 months, then they came back and scanned them again. They found decreases in cortical thickness in trans women between scan 2 and baseline, and those were most prominent in the medial prefrontal cortex. I should also state that they measured hormone levels and attempted to correlate changes in cortical thickness with hormone levels that they measured and did not find any correlations there. But instead of thinking about why that might be, they sort of concluded, oh, well, perhaps we just didn't have the most accurate measure of hormone levels. And if we had done, more measures over time, we would have found a relationship. But I want to offer a different interpretation of their findings. So we know that medial prefrontal cortical thickness is associated with perceived social support. So people who have reduced cortical thickness in this area tend to report not feeling as socially integrated or socially supported. We also know that medial prefrontal cortex cortical thickness, indicated here, is associated with a lot of psychiatric diagnoses and life experiences that are more prominent in trans women than they are in cisgender women. So PTSD, chronic stress, traumatic life events are all correlated with cortical thickness. And so what we have here is this failure to look at the first 6 months of transition from the perspective of a trans person. This is a time of enormous psychosocial change. People might have lost the support of their family and friends. And so I think it's important to think about that in terms of the social context of people's lives, rather than thinking, oh, everything has to be explained by estrogen. All right, now I'm going to talk a little bit about the resting state literature. So for folks who aren't familiar with resting state MRI, this is a method by which people were measuring correlations over time between regional blood oxygen level. People are just laying in the scanner passively. They're not instructed to do anything. And we're measuring blood oxygen levels and correlating them over time to identify parts of the brain that seem to be activating together. And the idea here is that they're functioning as a circuit. So one really sort of common criticism of the resting state literature is that we don't actually really know what people are doing unless we ask. So they might be thinking about their vacation, thinking about an argument they had, thinking about how loud the MRI machine is, that they're hungry, that their feet hurt. So we don't actually really know. But a lot of times when people talk about resting state, they talk about this default mode network, which is regions that collectively are more active at rest than during tasks. And the idea here is that this might represent some sort of ruminative or self-reflective function. So in terms of the resting state studies, again, same problem with lack of statistical correction and same problem with being, really underpowered. A lot of these studies tend to mix together trans people who are on HRT and people who are not. But by and large, they tend to find that the resting state connectivity profiles are more consistent with people's lived gender identity than their assigned sex at birth, with some exceptions. So I actually want to highlight this SPIES study now and talk a little bit more about it. So gender transition affects neural correlates of empathy, a resting state connectivity study. So in this study, I'm sorry, I just noticed some shocked faces. So in this study, it's a longitudinal study that involved a baseline resting state scan, the initiation of HRT, 4 weeks, a 4-week interval, then a second resting state scan, then a 4-month interval, and a third resting state scan. And this was all in, this included trans men and trans women. So if we just look at the baseline data, I think it's fairly obvious that the trans female group looks different from everyone else. And the authors choose to conclude from this that the cerebral organization of transgender people is different from cisgender people. I don't know necessarily that I would make that broad of a claim. It's clear that the cerebral organization of transgender men isn't different. And this is only one of the components that they measured. So I think that's a bit of a broad conclusion to make. Now let's look at the longitudinal data. They really focused here on the transgender women group because that was the group that was different at baseline. And they found basically an increase in connectivity, resting state connectivity over time, such that by the third scan, they were consistent with the connectivity weights of cisgender people. The sort of quote from the paper is that the resting state connectivity of transgender women normalizes over time, representing an assimilation to womanhood. They also looked at something called the emotional contagion scale, and they found that changes in connectivity between scans were correlated with performance on this emotional contagion scale for trans women. So they talk about the emotional contagion scale as a measure of empathy, but that's not actually what this measure is designed for. So this is a measure, the emotional contagion scale has people look at different face emotions and identify them. There are 4 different emotions present in the stimulus set: happy, fear, anger, and sadness. So it's really just looking at faces and identifying the emotion. And if we look at the trans data from this study, there's only actually a significant difference in their emotional contagion score at the final scan, and it's only for angry faces, such that trans women, after 4 months of transition, are much better at identifying angry faces. So the authors characterize this as trans women becoming more empathic as they transition. And I think that that's really being informed by this kind of essentialist and sexist idea that women are more naturally empathic and that must be related to estrogen in some way. But I don't think that that's what's happening here at all. We also know that this emotional contagion scale is most prominently associated with anxiety and threat sensitivity. And there's no sex difference in performance on this measure in cisgender people. So it's a bit strange to me to sort of argue that this is evidence that trans women become more assimilated, like more like other women as they transition. So this is a bit snarky, but I basically decided to fix this. I think that what's happening here is that as trans women begin to experience trans misogyny and unfortunately a lot of violence, that they become better at identifying people who might hurt them. And that's why we see this finding that's specific to angry faces and specific to social threat. So biological reductionism is a bad idea. And I think what a lot of these papers do is they make leaps from things like genes and environment, things like brain structure and function, to complex social and cultural identities. Being trans is a very complex social and cultural identity that's subject to a lot of hierarchies. It results in differences in access to resources. And so equating any observed biological features that are different between trans people and cis people with some sort of innate at birth difference is really sloppy. You know, of course, all of these different factors are interacting with each other in really complicated ways. And so I think if we're going to do this work well, we really need to start thinking about the developmental and social context of transgender people as whole human beings, not as just sort of just thinking about us in terms of our hormones. So I wanted to end with a couple studies that I really like. So this is a study where transgender children and a matched sample of cisgender children were asked to do an impossible computer task. And while they were trying to do this impossible computer task, there was a recording of someone ridiculing them playing in their ear. So it's pretty mean study design. And so what they found were they found differences in the trans youth and the cis youth in terms of both baseline stress levels and stress reactivity, and also how they reacted to the social stressor. So this graph here, we see the negative mood score on the y-axis. So a higher score indicates more negative mood. And then on the x-axis, we have the time that the sample was collected. And we see for the trans kids shown here in black that they show a elevation pretty rapidly over the course of the stressor. So I should clarify, Z1 here is baseline. So basically, as soon as the stressor starts, they have this really elevated, the pretty dramatic uptick in negative mood. But also just at baseline, trans youth had lower basal cortisol. So when we see lower cortisol levels at baseline, we typically think of that as being associated with, a chronic stress effect that's dysregulated HPA axis function. They also showed greater GSR at baseline. So that would indicate more sympathetic activity, more arousal in the trans kids just at baseline before they've even started the stressor. And higher baseline feelings of lack of control for the trans kids. And so I think when we start doing research, we need to think about just the baseline conditions for trans people being really different in terms of autonomic nervous system function. And think about that in our study design, and think about that in terms of, what we think our stressor is doing in terms of modulating these systems. So, and then finally, the trans kids also reported greater increase in negative emotion and loss of control during the social stressor. They found it to be much more stressful. Okay, so I'm gonna talk now a little bit about an fMRI paper. So as far as I know, this is the only fMRI paper that's looked at the social context of trans people. So this is a task-based fMRI study of transgender people, transgender men and transgender women, and cisgender men and cisgender women. And for those that aren't familiar, they used a social exclusion task called the cyberball task. So prior to going into the scanner, the research participant meets two other people who are actually, there's a bit of deception in the task. So once they go in, they're told, during this task, you're going to play pass with the two people that you just met outside of the scanner. That's all they're told. But they're actually, of course, not playing with real people. They're playing against two computer figures. And this is basically what that task looks like. It's a little boring, but basically they're playing pass. And for one portion of the task, they're being included equally. So they're being passed the ball just as much as the other players. And so this is the inclusion portion of the task. For the social exclusion portion of the task, the two computer players just pass to each other and completely exclude the research participant. And then for the third phase, the participant is re-included into the game. So this is what they found. I don't want you to worry too much. There's a lot going on in this figure, but I do want to specifically highlight this ventral ACC finding. So basically what they found here, these are plots of the extracted signal of the ventral ACC during the period where the participant has already been excluded and now they're being re-included. And so we see here the cisgender men, cisgender women, trans men, and trans women. And there's significant group differences between both trans groups relative to the cisgender group. And the authors conclude from this that there are medial prefrontal differences that occurred during the re-inclusion portion, basically such that trans people show a neural signature associated with social exclusion even after they've been re-included into the game. So basically, this idea that it takes longer to sort of get over the social exclusion component. Now, obviously, there's a little bit of hand waving there. It's always, you know, we're looking at correlations in fMRI always. But I think this is a really interesting way of thinking about how the chronic effects of social exclusion on trans people and how this might show up in our brains. Importantly, this ventral ACC finding shows up in our pathophysiological models for mood disorders quite a bit. And it's a region that actually shows up in the transgender literature quite a bit in terms of being different from cisgender people. But I think this study really gives us a lot more context into why that might be. So all of that said, how can we do a better job? So the first thing that I would say is that we really need to partner with transgender communities. A lot of the pitfalls here can be avoided if we're partnering with the communities and get a sense of what community priorities are, what's going to be the most helpful to them, and also, you know, have more insight into the social world that trans people are navigating. Another reason that it's important to do community-based recruitment. Most of these studies recruit transgender people from gender identity clinics. So there's a number of reasons why that's problematic. First of all, gender identity clinics tend on average not to really be representative of the demographics of the community. They tend to be much more binary identified and tend to be higher SES and mostly white. Another reason why I don't think this is a good idea is that for the handful of studies that did actually exclude for psychiatric diagnoses, they used a psychiatric evaluation that was done at intake at the gender identity clinic. So this was the sort of screening for psychiatric diagnoses that was done prior when a trans person is going in and being assessed for access to hormones. And I think any trans person will tell you that access to medical transition is really heavily gate kept. So the WPATH Standards of Care 7, so World Professional Association for Transgender Health, they write the standards of care for transgender health. And according to the most recent standards of care, mental health concerns must be reasonably well controlled before a transgender person is allowed access to hormones. And so what this means in the context of being in one of these gender clinics is that transgender people have to walk this line whereby they're distressed enough that they should get access to the hormones, but not so distressed that they are denied access to the hormones on the basis of being you know, unstable in some way or not having their mental health under control. And so when we recruit in this context, I don't think we're getting good data about people's mental health symptoms. And I'm really skeptical of a study that's recruiting from a gender clinic using the psychiatric screening done by the clinician who's going to be prescribing hormones, and then claim that they have this sample of trans people with no history of depression or anxiety. That's just really not consistent with the community literature that we have that shows, like I said earlier, 90% lifetime prevalence of major depressive episode, 40% lifetime prevalence of suicide attempt or suicidal ideation. So I think this is a really big problem when we don't recruit from the community. And I should also say that there's actually been several studies published recently in the transgender health literature that talk about how transgender people navigate this situation. And all of these studies basically conclude that trans people, because of the way that the access to HRT is set up, trans people really can't be honest with their providers about their mental health concerns. So of course, this is a problem for research, but most importantly, this is a problem for our community. A lot of these studies don't really match and compare groups based on both gender and sex assigned at birth. They typically choose one or the other. And I think it's really important that we include all combinations of sex and gender as comparison groups. I think in terms of study design, we need to think carefully about what our hypothesis is and if it makes the most sense for our comparison group to be matched for gender or for sex at birth. We also should be thinking about including a cisgender control group that's matched for depression and anxiety symptoms. I recently had an RA who was doing recruitment for another lab study approach me for some advice because they had recently started collecting gender identity and sex assigned at birth data for their depression study. And they were finding a lot of trans people were participating, but all of them were in the depressed group. None of them, they didn't find any trans people in the sort of control group. And so she was reaching out to me to see if I could help her find a trans control group that didn't have any history of depression. And that group is just not, it's just not representative of the community. And so I think the best solution then, instead of trying to find that small 10% of trans people with no history of depression, is to instead really match for mental health concerns. We also need to be thinking about assessments for stress, trauma, and the social changes that come with transition. And I think I've really kind of hit that point already. Another thing that I want to mention too is that these studies often take a great deal of time and effort to characterize and diagnose the trans group with gender dysphoria according to DSM-5 criteria. But they do all of this work to identify trans people with gender dysphoria. But then instead of looking at gender dysphoria, which is a diagnosis based on it's a continuous variable, it's a symptom, right? Instead of looking at that, they then just say, oh, well, that's the trans group. So they take what could really be a nuanced continuous variable, which is probably more proximal to our biology than a binary social identity, cis versus trans. So I think I would really like to see gender dysphoria measured continuously and then look at that in terms of brain structure and function. Not only is it more proximal, gender dysphoria is an actual clinical condition that causes distress for, a lot of trans people. And so looking at the neural correlates of that is actually, I think, has the potential to be much more useful for trans people than, sort of just a binary trans versus cis comparison. So I've included here a example of a two-step method for collecting demographic information. There's a couple different ways you can do this. So here's one method. You have two questions. The first one is, do you consider yourself trans? And then the second one is, does your gender match the gender you were assigned at birth? Another method, and this is the one that we're using at Pitt now for a lot of our studies, is what is your gender identity? And then you have the option to choose multiple multiple answers. And then the second question is, what is your assigned gender at birth? Meaning the gender marker which appears on your birth certificate. And this, this bottom two-step method in particular has actually been well validated in clinical and research settings, and in samples of people across all age ranges. Both cisgender and transgender people are able to understand this question and answer it accurately. So, you know, I think either of these options would be a good option moving forward. You know, I think we do actually need information about people's sex assigned at birth because we are studying biology, but I think it's really important that we get, you know, both pieces of information, even in our studies where we're not looking at transness specifically. I can tell you in one of my colleagues' R01s, they had an R01 looking at depression. They asked about sex just as a binary, and they didn't have any trans people in their sample. The R01 got renewed, so same exact recruitment methods, same exact population, but this time they changed the form to the two-step method, and now 10% of their sample is trans. So, you know, my argument here is, wouldn't you rather have that information as a researcher? Another sort of a really important point is that even our categories of sex and gender are still really insufficient to get at a lot of the constructs that I think we're really interested in as biology. So this is a really great short paper in Science, all trans feminine authorship team. And in it, they are talking, and they all do rodent work. And in it, they talk about sex as a construct. Not that it's not real, but that it's actually composed of a lot of different factors, right? We have karyotype, hormone levels, we have anogenital different distance in rodents. And so they talk about, think about what your hypotheses are and think about what component of the sex construct is most appropriate for you to measure. And finally, I just want to end, shameless plug for an upcoming special issue of Biological Psychiatry Cine that is a diversity issue. And this is a figure that I created to talk a little bit about how we can do research in a community-informed way. And I think it's really important that we do this at every step. So the first thing before we even start is we need to have a little bit of baseline familiarity with LGBT culture, and particularly how it might relate to psychiatry. There's a really long and contentious history between trans people and psychiatry. Psychiatry has really harmed a lot of trans people and continues to do so even today. And so, you know, if we're coming at this as psychiatrists or as people who are researching mental health, we need to know a little bit about that history, because that's going to help us understand the perspectives of community members who might initially be very mistrustful. In terms of hypothesis generation and model building, I think it's important to think from a minority stress and a developmental approach. We need to be thinking about not just trans people in the context of the present, but also the neurodevelopmental context in which they're growing up, by and large, in the closet, and how that might affect their mental health. As I've already sort of mentioned, doing community-based participant recruitment, another sort of consideration, particularly in studies of trans youth, is thinking about how you're describing the study on all study materials, including parental consent forms. You don't want to accidentally out someone to their parents. Data collection, thinking about how we're asking about demographics. A really sort of frightening thing to start thinking about is all of the methods that we use have been validated pretty much exclusively for cisgender people. So we don't know necessarily about the item validity or the, sort of usefulness of a lot of the assessment tools that we use in a trans population. So that's a really important thing to think about, especially if we're looking at a measure that has sex differences. If it's normed by sex, it becomes really hard to know what to do with that in the context of a trans population. I think I've really hit the comparison groups and psychosocial factors issue quite a bit already. And then given the current climate for trans people right now, I think we need to think really carefully about how we talk about our findings. It's clear that trans people, especially trans youth, are being targeted right now by a lot of really harmful and just horrible legislation. And so if we find differences between trans people and cis people in our research, we need to be prepared to talk about that to the public in an accessible way that doesn't perpetuate harm against a already very marginalized community. So that's all I have. I'm happy to take any questions. I wanted to give a special thanks to Laura and Rachel, who are currently co-authors on a review that we've written together about this literature, and my funders. I also wanted to highlight my Twitter. I recently just pinned a survey, a climate survey for LGBT neuroscientists. So if you're an LGBT neuroscientist, please take the survey. And that's all I have. So thank you.
Thank you so much for an excellent talk. Yeah, anyone who has questions, either raise your hand or unmute yourself and ask away. Daniela.
Hi, yeah, thank you. It's
Marina asked a question. I'm sorry, Daniela, go ahead. And then maybe Urena can go. She asked a question in chat, but go ahead.
Oh, okay. All right, so just wondering, what do the participants themselves know about the purpose of the experiment? What are they being told? What are the ethical committees are instructing them? I mean, I assume there's a lot of variability. I was just wondering if you looked at that issue.
That's a great question. I don't know, unfortunately. And I think, for those of us who are researchers, I don't know. I think it's easy for us to forget how intimidating the sort of process of participating in research could be for someone who isn't familiar with it. So I don't know what the participants in these studies were told, but I think, it is worth really taking some extra care during the consent process with trans people. I will say that there was recently a study at UCLA that was shut down. This was a study, a neuroimaging study of trans people, where they were showing transgender people pictures of their body pre-transition and looking at their response to it. And of course, it was incredibly distressing to trans people. And so it actually got shut down on ethical grounds after a whole lot of local community outrage and complaint to the IRB. So, I think, yeah, the short answer is I don't know what these people, what the participants were told, unfortunately.
Thank you.
Well, this might be a good time for me to jump in with my question, if that's all right.
Sure.
So we know that, particularly in neuroscience, but across all of biology, we know that sex differences are largely overlapping distributions of traits, where any one trait has very low predictability for any given person and or any given arrangement of other sex-related traits. And this seems to be crucially true in neuroscience, in that a lot of research by like Daphne Joel has shown that there's more such a mosaic of sex-related traits, which at some point it's even kind of questionable whether or not that's relevant. And so what I sort of wanted to ask is that these sort of like perceived biological differences between cis men and cis women and between trans people and cis people are being used to exclude trans people from public life by various politicians and lawmakers. And so I wanted to ask what you think sort of can be the impact of looking for neural correlates of either sex or gender identity or trans status, and any particular dangers you feel are associated with that.
Yeah, so I actually have thank you so much. That's a great question. So this idea of there being distributions of sort of sex typical traits, but there's actually much more variation within group and than there is between groups. That, you know, all of these sorts of sex differences are really, you know, not great at predicting an individual's sex. And so, you know, what is there a way to do this that's in service of trans people? I will first say, I actually have a file drawer study where I looked at the effects of a lecture on the biological basis of sex on transphobia in a sample of cisgender undergrads. And so I gave them a transphobia scale before they had that lecture and then after. And what I found was that the lecture served to polarize whatever beliefs they had at baseline. So the people who were really transphobic at baseline became more transphobic after this lecture, and the people who were not became less so after this lecture. So I think the danger with these sorts of arguments is that they can be used as evidence for whatever political opinion you already have, or whatever bias you already have. And that's because, you know, if there's a biological difference, it's very easy to sort of say, oh, well, that indicates, you know, some sort of pathology, right? So in terms of ways of doing this that would actually be in service of trans people, I think I would really like to see studies that focus a little bit more on the neural correlates of depression in trans people, because it is such a pervasive problem in our community. And just from a neurodevelopmental perspective, I think it's pretty clear that the high rates of depression in our community are because of neurodevelopmental stressors, ongoing stressors, trauma, marginalization, et cetera. And so, you know, understanding the neural correlates of depression and the pathophysiological method there can be really helpful to trans people, but also to a whole lot of people. And, you know, it may be that there are different targets for intervention or treatment for people who have depression that might be more secondary to that sort of social marginalization. And no one's really looked at that. So I'd like to see someone should do that. Does that answer your question? Okay.
Yes, definitely. There's a question in the chat here that I can.
Yeah, and do Urena or Chris want to unmute themselves and ask a question? Or Dr. Edmiston, maybe you could just read the question and answer it.
Okay.
I'm happy to read it as well. I think Urena had.
Oh, here's Urena raising her hand. Good, Urena, go ahead.
Hi, good afternoon, everyone. So, you know, I applaud you for doing such impressive work, but I also know that there are some challenges in conducting this type of research. So, you know, I'm thinking about how difficult it can be to conduct studies that to some degree question established cultural norms and assumptions. And so I'm wondering if you can speak to how we can overcome some of these challenges, perhaps at the funding or institutional level, thinking about maybe NIH or, you know, working with IRBs, et cetera.
Okay, well, that is a huge question. Thank you. Thank you for asking it. So I want to make sure I'm understanding. You're sort of saying, like, how can we engage with institutions to avoid some of these problems related to like ciscentrism, basically?
Yes, it is a broad scope question. You know, sometimes I find that funding institutions may not be fully understanding why studies outside of the expected norms are important, what is their impact, and also perhaps working with institutional organizations like IRBs, they may have similar challenges trying to contextualize this type of work. And so I'm wondering if you can speak to any of those challenges, how they may have impacted you, how we can overcome them, et cetera.
Yeah, so I guess I will just say straight off the bat, in terms of how things have impacted me, I haven't been able to publish any of my trans-related work. I have trans papers that have been sitting on my desk for 7 years. And that's because this is the model, this model of group differences, and one is pathologized and the other is not. So, you know, I'm pretty ambivalent about, just because it's very close to me. So you know, it's very frustrating. I'll just be very honest. In terms of what we can do institutionally, I think part of the problem is that all of our funding mechanisms are really based on a, you know, especially NIH, it's very much like a sort of pathology lens, right? And so how do we, you know, obtain funding and design studies that aren't really focused on, you know, pathologizing one group? So I think that's one concern. I think in terms of IRBs, I think that there's probably there's a lot of training that can be done. Unfortunately, I think a lot of times this training is left as a service activity for the most marginalized people in the department. And so I think that there needs to be institutional support to hire people to do audits and trainings for for our IRBs in terms of, you know, diversity issues. You know, I think just more mechanisms that reward paradigm shifting research or innovative research. I have a feeling that people who want to do this type of work in a more nuanced way might have better luck with like a foundation grant. I think that, you know, the model at NIH right now, it just makes it difficult, I think, to do this work through an NIH funding mechanism. But I think that that's going to change. I think it's already changing. I saw there was recently an RFA put out for research, for our grant mechanisms for research on mental health outcomes related to medical transition for trans people. So I think that there is interest at the NIH, but yeah, I think it's an uphill battle for sure. I don't know if that answers your question. I mean, I think your question is one that I'm constantly asking myself, so I don't necessarily have like an easy answer. Like you might know better than I do. So I
wish.
Yeah.
No, that was really thoughtful and insightful. Thank you. I agree. I face similar questions probably on a daily basis. So I appreciate your reflections. Thank you.
Thank you.
Should we answer Chris's question? And
yeah, if you don't mind, maybe answer Chris's question.
Yeah,
I can read it all. Okay.
Yeah, go ahead.
Chris says, thank you for this important talk. You spoke a little to this when discussing sex as a construct, but would like to hear your thoughts regarding studies assigning groups with regard to sex assigned at birth, given that sex at birth is typically assigned based on how close external phenotypic traits appear to assumed binary sex categories, as opposed to chromosomes, internal organs, and other ways of assigning sex.
Yeah, so I definitely, I cannot more strongly recommend the science paper that I had highlighted. They talk about this. I think it's challenging, right? Because, you know, for me, I'm not necessarily particularly interested in sex differences in my research. And so it's challenging. You know, for our grants, we have to write a sex as a biological variable paragraph. And I'm always sort of like, well, how much do I want to push the envelope on this? You know, especially since sex differences is not typically, at least not in any of the work I've done so far, not like my primary outcome of interest. But I think for people who are interested in looking at sex differences, you're absolutely right. Like this idea of looking at the different component constructs or the different components of the construct of sex based on sort of what your a priori hypotheses are about mechanisms, right? We need to start thinking about mechanisms rather than these sort of broad binary social categories. I think that just like the air we breathe is so much informed by this cis-centric idea of there being men and women, and men have these types of bodies and women have these types of bodies. And even for us as biologists, we've overlooked this. So I think, depending on your hypotheses, you might want to look and measure some of these different constructs. In terms of how to analyze that data, then I think it gets complicated, right? We might need to put our heads together and start brainstorming different ways of looking at this. I know the aforementioned collaborators study that I had talked about, where they started collecting sex assigned to birth and gender data. Well, now that we have that information, what do we do with it? How do we include it in our analyses? What if our trans group overall looks completely different from all the cisgender people in terms of, clinical characteristics? What does that mean? I feel like everyone's asking me questions that I just sort of, I can't give like a definitive answer to. It's more like, yes, that's a great question. I don't know. But I guess, yes, there, I think we just need to think in a lot more nuance. Yeah. Thank you, Chris.
Any more questions? Great. Well, I suppose it's time to give Dr. Edmiston another round of applause. And there's a meet and greet with students in another Zoom link that should have been in the same email directly after this.
Thank you so much for joining us. We really appreciate it.
Thank you for having me.